Hello everyone. So the aim of our practical is to find out the RBC count of our own blood. The requirements are the Newbar's chamber, the RBC dilution pipette, Thomas Coverslip, a watch glass, hayems dilution fluid for RBC dilution, 70% alcohol, xylol, some cotton, a pricking needle, and lastly a compound microscope. The principle of the practical is that the number of red cells in the blood are too many but the size of them are too small. It is impossible for us to count the cells even under high power microscopes. So this difficulty is overcome by diluting the blood with a suitable dilution fluid. The diluted blood is then placed in between the counting chamber and cover slip. The number of the cells can be calculated by multiplying the number with the dilution factor. The procedure starts by cleaning the counting chamber and the lenses of the microscope with the help of xylol and absorbent cotton. The counting chamber is adjusted and observed for RBC squares under the low power of microscope. The ring finger is sterilized with the help of alcohol and it is pricked boldly with the help of a pricking needle. A droplet of blood is formed and the blood is sucked up in the RBC pipette up to the mark 0.5. But make sure that there are no air bubbles for accurate results. Now we can clearly see the blood in the RBC pipette up to the mark 0.5. Immediately the RBC dilution fluid is sucked up to the mark 101. The pipette is brought to a horizontal position and a simple knot is given to the rubber tube. The pipette is rolled between the palms to mix the blood with the dilution fluid for about 1 minute. The first drop is discarded and then the pipette is held at an angle of 45 degrees and a drop is allowed to come out from the pipette. The RBC chamber is located and the RBCs are counted in the 16 smallest squares. This is repeated to complete the total RBC count in 5 chambers, namely A, B, C, D and E. Here we have the dimensions of the squares and the equation to find the total number of RBCs in the undiluted blood, where N is the total number of RBCs in the 80 smallest squares. When we add the total number of RBCs in the chambers A, B, C, D and E, we get our N to be around 518. When we put the value of N in our equation, we get the total number of RBCs to be 5.18 million. The normal RBC count in male is taken around 5 million and in female as 4 million. Thus, we can say that our blood is in the normal range of RBC count.